Hello students, welcome you all on my channel. In this video I am going to discuss about the physiology of excretion, which is the most vital part of excretory system. In this heading there are two processes. The first one is ornithine cycle, that is to say formation of urea in the liver, and the second one is urine formation, which is taking place in the kidneys. So the first one is ornithine cycle. This cycle is also known as urea cycle or Krebs-Hensleit cycle. Krebs-Hensleit cycle. So we are calling the most convenient is the ornithine cycle instead of calling Krebs-Hensleit cycle, or simply you may call urea cycle. In this process there are formation and transformation of three amino acids, namely ornithine, citrulline and arginine, for the repetitive cyclic process of the ornithine cycle. With the help of a schematic diagram, I am going to demonstrate the ornithine cycle. Here, CO2 combines with ammonium to form carbamoyl phosphate. And this carbamoyl phosphate is formed in the presence of an enzyme. It is known as CPS first. CPS means carbamoyl phosphate synthetase first. That I will write later, that to complete this cycle. Now the carbamoyl phosphate combines with ornithine cycle with ornithine amino acid, and then forms another product called citrulline. So here I am going to write here ornithine or ornithine. Now here another enzyme will work. When carbamoyl phosphate combines with ornithine to yield citrulline, one enzyme play vital role. That enzyme is called OTC. The another enzyme, OTC an enzyme result in the formation of citrulline in the presence of ornithine amino acid. So OTC. Now here citrulline is formed. And citrulline is also amino acid. Citrulline now combines with aspartic acid. Aspartic acid is combining here. Aspartic acid, or you may simply call it aspartate. When the aspartic acid combines with citrulline, it forms argininosuccinic acid. It forms argininosuccinic acid. But here another enzyme will work. and that enzyme is known as argininosuccinate synthetase. As you know, the suffix ASC depicting the enzymes here. Now it splits into arginosuccinic acid splits into fumarate and arginine. Fumarate and arginine. So here the fumarate will be eliminated, or fumaric acid will be eliminated from the body. Fumaric acid, or simply fumarate, is eliminated from the body. Now here the arginine will form. Arginine will further break into urea and ornithine. So here arginine, now it splits into urea plus H2O, and this arginine yields into ornithine, and thus the cyclic process is continuing. Here it breaks into arginosuccinic acid, it breaks into fumaric acid or fumarate and arginine, and this arginine will further break into urea and H2O. Now here one more enzyme is needed, and that enzyme is known as arginase. Arginase. In the presence of arginase, arginine is converted into ornithine, and then ornithine participate in the cyclic process of the urea synthesis. Now here I told you that ornithine, number one, it is the first, this is the second, and this one is the third. 3 amino acids are formed and transformed. Why I am using the term transform? Because of one amino acid is participating in the reaction, then forming another amino acid, then another amino acid participating in the reaction with the help of enzymes, and then converting into the product, and then again the product is breaks into the amino acid. So this is the cyclic process. It is continuously going in the liver cells. So urea is synthesized in the liver, and this is hampered when there is diseases related to the liver. In cirrhosis, this cycle is hampered. Cirrhosis means liver cancer. In the liver cancer case, this cycle is hampered. So it is ornithine cycle that I have mentioned here. So now the time is to write the enzyme's name. The first enzyme is CPS. Its full form is carbamoyl phosphate synthetase. The second one is OTC. It's an enzyme. Ornithine transcarbamylase. See, again I am explaining you, but before that I want to split this. I told you this participates in the two parts of the cell, one in the mitochondria and other in the cytoplasm. This portion, when the CO2 combines with ammonium in the presence of an enzyme CPS first, it forms carbamoyl phosphate. This participates in mitochondria. This reaction, this side, and this reaction, the cyclic process where the amino acids are forming and transforming another amino acid, this takes place in the cytoplasm of the cell. So you must have understand, and I hope you have understood it clearly about the ornithine cycle. Ornithine cycle is very vital. It requires few steps, and namely the three enzymes, CPS, OTC, and arginsuccinate synthetase participate in the up to formation of the arginosuccinic acid. After that, arginosuccinic acid breaks into two, fumaric acid or fumarate, that will be eliminated from the body. Then arginine combines with arginase enzyme and yields ornithine and splits into urea and water. Urea and water is eliminated from the body by the filtration mechanism of filtration mechanism of kidney, and this is ultimately results in elimination. Here the hepatic vein carry a rich amount of urea and takes to the kidney for the filtration. The renal vein contain least amount of urea because the blood is filtered and now blood contain. So students, I just told you about the urea and H2O. This urea is eliminated along with the water, and the remaining part of the amino acid, this ornithine, will participate in the further reaction, and this cyclic process will be continuous in the liver cells. So as you know, this CO2 combines with ammonium with the help of an enzyme CPS to yield carbamoyl phosphate. Carbamoyl phosphate combines with ornithine in the presence of OTC, that is the ornithine transcarbamylase enzyme, and forms citrulline, the second amino acid. The citrulline combines with aspartic acid or simply aspartate in the presence of arginosuccinate synthetase. This process will takes place in the cytoplasm, and to yield arginosuccinic acid, arginosuccinic acid breaks into fumaric acid or simply fumarate and arginine. Again, this is the amino acid. This amino acid is splits into urea and H2O. In the presence of arginase enzyme, it forms ornithine, and this cycle will be continuous. So students, that's all for this video. Thank you for watching. Stay tuned, and in the case of doubt, you may ask me. Thank you, students.